Good afternoon, everyone, and welcome to the next edition, this current edition of Live from the Laboratory. Can I have the slides, please? We're going to hear today about Parkinson's disease. First, I'll introduce myself. I'm Mitchell Kronenberg. I'm the President and Chief Scientific Officer. And then we'll hear from Dr. Alessandro or Alex Sette, who's a long-standing, highly productive professor here. And he'll give us some research updates and background on Parkinson's. And then we'll hear from Cecilia Lindestam Arlehamn, who's also on our faculty and has recently received an important Parkinson's disease research grant. And then we'll hear from Steve Wilson at the end to talk about how our supporters have positively impacted us. And finally, there'll be question and answer moderated by Steve Wilson. Next, please. A little background about us. We are a nonprofit research institute. We were founded in 1988. We have almost 500 employees. And we're organized into independent laboratories led by principal investigators. But we also have a very large number of highly educated people, technicians and support staff, postdoctoral fellows, and other trainees, all working together to advance knowledge in immunology. Our mission is to engage in a world-class research program with a focus on the immune system. And what we will conduct, share, and partner such that the results of our discovery will make outstanding contributions to the betterment of human health. Next, please. Given the coronavirus pandemic, our faculty, in fact, have organized themselves into a task force to better understand the immune response to this virus so we can develop therapies. Next, please. And I just want to highlight one finding from this task force that was published in the journal Cell. You can see that in the letters on the right. And this is from Drs. Crotty, Sette, Weisskopf, and Grifoni. And what they found is that most people who are exposed to the virus make a good immune response. The virus is not immune suppressive. And also that there is some prior existing T-cell immunity to this virus, probably based on circulating corona cold viruses, which they later showed was in fact the case in a subsequent paper. So this kind of work has really focused enormous attention on immunology and our work in particular. Next, please. And in fact, I was really quite amazed to see on CNN, which I know you all watch for many hours a day, a congressman asking Tony Fauci about T cells, and Dr. Anthony Fauci, the director of National Institute of Allergy and Infectious Disease, holding up our paper, meaning Shane Crotty's and Alex Sette's paper from La Jolla, and citing the important work from La Jolla on T cells. We are very proud of that, of course. Next, please. So we have a very clear and powerful focus. And we're an immunology-focused organization because studies of the immune system provide the greatest opportunity for affecting human health. And that's why we've adopted the motto, life without disease, because so many diseases are caused by misfunctioning in the immune system or can be prevented by influencing or boosting the immune system. And if you look on your right, so many different areas, allergy and asthma, autoimmune disease, cancer, chronic inflammation, vaccine discovery, and of course, infectious disease, which we've been talking about. But today, we have a different topic, Parkinson's, a disease of the brain. And many people used to think that the immune system and the brain were quite separate, but that's not the case, as you'll hear later in this talk. Next, please. So we've organized our research into disease research centers, infectious disease and vaccines, of course, cancer and cancer immunotherapy, and autoimmunity and inflammation. So we might think of this as a three-legged stool. And I can assure you that immunology is fundamentally about this immune system knowing when and to respond and when not to respond. And all of these areas are in fact interrelated. Next, please. So in fact, our lab heads, although we've assigned them into one or two of these areas, in fact, discoveries about when the immune system knows to turn on, which kind of cells, what kind of response. Somebody working in cancer can have important insights into infectious disease or vice versa. Next, please. So thank you for listening to that little overview and introduction to the La Jolla Institute for Immunology. And next, I'm going to hand the platform, the virtual platform, if you will, over to Alex Sette, who is truly one of the leading researchers on T cells and infectious disease and other conditions, inflammatory disease, as we'll hear today, in the world. Alex, please.
Hello, everybody. Okay, yes. Hello, everybody. Let me start and share my screen. Okay, yeah, so thank you, Mitch, for a nice introduction. And let's jump in and discuss a little bit, introduce what is Parkinson. And Parkinson is a very important neurodegenerative disease. It goes in this group of diseases that, such as Alzheimer or ALS, that have to do with neurodegenerative process in the brain. It's a very important disease, affects 6 million people worldwide and 1 million people in the US. And the general importance of this disease is growing over time, also because in general they are associated with a later stage in life. In particular, Parkinson has to do with the degeneration of the substantia nigra, the black matter in Latin. Black matter is because the neurons that are in the substantia nigra are black because they make dopamine, which is an important molecule that the nervous system used to communicate. And in the case of Parkinson, as you will hear also later on from Cecilia, these neurons are basically gone. They degenerate and they die and they are absent, and the functioning of the brain is impaired. So what is going on? There are a variety of different symptoms, but in Parkinson, there's essentially two main categories. One are motor symptoms, so basically shaking, rigidity, and other things like that. But then there are also non-motor symptoms, symptoms that don't have anything to do with the movement and motor function, but has to do with other parts and functioning, constipation, cognitive issues, sleep disturbances, loss of sense of smell, and so forth. So it's a very complicated clinical picture. And it's really a pressing issue because in reality there is no real conclusive laboratory test. So you can't go to a lab and they run something on your blood and say, yes, you have Parkinson's or you don't have. The diagnosis is complex, and it's also largely by exclusion of other things that could resemble Parkinson. And there is no cure. There are interventions that manage symptoms, but there is no conclusive cure. So let's take a step back, and Parkinson is to be seen also in the broader context of inflammation. The inflammation is essentially a sign that the immune system is in action. It's caused by cells of the immune system. So this is a normal process. It's actually very important because it's how the immune system gets rid of microbes and other enemies, so to speak. But there is also a dark side of the inflammation because it can also cause disease, can cause tissue damage, like, I don't know, in rheumatoid arthritis, for example. And also you can have chronic inflammation, which is usually detrimental. So when things are in balance and there is the right amount of inflammation, everything is good. But you can have pathological conditions where there is, in general, a suppression of inflammation or immune activation. And for example, you may have a situation where you don't detect mutations and destroy cancer cells when they're originating, or you can have chronic infection. Or on the other side, you can have too much inflammation, such as, for example, in the case of allergies, you react against pollen, but that's really not necessary. Or in autoimmunity, where the immune system attacks your joints, and so you get rheumatoid arthritis, or your pancreas and get diabetes and so forth. So it was really not appreciated that Parkinson had an autoimmune component. But when you look a little bit deeper into the general disease process and symptoms and biochemical events that are associated with Parkinson, what you see is that in Parkinson, there are these Lewy bodies, which are basically dark clumps of a particular protein. It's called alpha-synuclein. And this is very characteristic of a disease. And this has an interaction with this particular type of cells called microglial cells. And these microglial cells are a type of cell which are from the general family of macrophages or monocytes. Basically what these cells do, they're scavengers. They go around and they see clumps and aggregates and they get rid of it throughout the body, all these different types of monocyte or macrophages. That's what they do. So this is important because this was the first hypothesis. And we thought, well, maybe what's happening is that some pieces of this synuclein end up activating T cells and then activates an inflammation. In fact, that's 2 years ago, we published, Cecilia and I, a landmark paper where we actually showed for the first time that that was the case, that Parkinson's disease patients actually had T cells that recognize little fragments of this protein, alpha-synuclein. So what we think is going on is that the microglia recognizes and eats up these aggregates, these clumps of synuclein, and then pieces of this half-destroyed clumps actually make it up on the surface of these microglial cells, and T cells recognize this. That's what they do for a living. They recognize pieces of proteins on the surface of different cells that should not be there. So they get activated and they cause an inflammation. And Cecilia will go in much more detail about the complex dynamics that we have been uncovering throughout this last few years. In closing, I just wanted to go back to a potential connection with COVID, which is totally highly speculative at this point, but it goes into a general context of, so if Parkinson and neurodegenerative diseases are due to basically an unwanted or unhealthy inflammation, what can trigger this inflammation? And there's been a lot of speculation throughout the years that this may have to do somehow with viral infections. So that the idea is that you have a viral infection that somehow is connected to the brain, and this induces an antiviral response, and this causes an inflammation locally, and this inflammation then triggers an autoimmune attack, which then eventually result in loss of the neurons. So this is one area where we are very interested in, and we're looking at really the interaction between infection and neurodegenerative disease, which is really an area where there is very, very little known. And I want to go back for a second and lead you to a speculation here in terms of the data that Mitch was referring to earlier. So we had this study in Cell where we showed reactivity against common cold corona in unexposed individuals, and that this reactivity in common cold corona actually led to cross-reactivity, recognition of SARS-CoV-2 fragment, which are similar. Some fragments are similar in structure to the fragments that are contained in common cold corona. So that led to the speculation, which I emphasize at this point is still complete speculation, that basically if you have had recent or heavy exposure to common cold coronavirus, this cross-reactive T cells, with T cells that can cross-recognize both common cold corona and SARS-CoV-2, may help inducing maybe a faster or stronger immune response, and may translate in an advantage, in a reduced clinical severity of disease. And there are precedents for that. For example, in the case of influenza, where it was shown in the last swine origin flu pandemic, but that was indeed the case, that people that were had some cross-reactive immunity still got infected, but had a somewhat less severe disease. So what that has to do with Parkinson's, we don't know. But one thing that is apparent is that there is an interaction. There's some old data that showed that they reported that people with Parkinson had a higher level of exposure to common cold corona. So we're very interested in actually understanding whether that has anything to do with COVID susceptibility. It could be that people with Parkinson actually are relatively more, have lesser disease symptoms or severity than non-Parkinson. It could be the other way around. So this is one area that we are very actively looking into. And in the general area, what is the relationship between having Parkinson and having general immunity to other things like viruses or bacteria? And I'm gonna now pass it on to Cecilia.
Thank you, Alex.
How do I?
And hi, everyone. I really appreciate you taking time to sign in and listen to this today. I will talk to you more about the development of Parkinson's, which is a very long and gradual progress. So in the initial risk phase, the majority of the neurons that Alex was mentioning are still remaining. As you move into a sort of preclinical phase, neurodegeneration starts to happening, which is represented by this line here. And in what's called in the Parkinson's field as the prodromal phase, there's more neurodegeneration and also these non-motor symptoms that starts to happen, such as loss of smell, constipation, sleep disturbances. But those symptoms can be due to a long range of things and not necessarily only Parkinson's. So usually individuals are only diagnosed when they start having their motor signs. That's when you enter a phase of clinical Parkinson's disease. And at that time, the majority of the neurons are lost. And when the neurons are lost, they're not coming back. So we're really interested in the entire Parkinson's field to explore a biomarker for early detection, something that can be used here to detect the disease and delay or prevent this neural degeneration. And as Alex mentioned, we published a study back in 2017 where we see that T cells from patients with Parkinson's disease recognize peptides or targets from this particular protein, alpha-synuclein. And now we want to go on and understand the prodromal phase, and could these T cells potentially be useful as a potential disease and prodrome marker? So we really want to look at longitudinal changes in alpha-synuclein-specific T cell reactivity. And we were lucky to have access to an individual's frozen down cell samples that were collected in this phase, which can last for a long period of time, over 10 years, and also in this phase, which is the clinical phase. And when we look at reactivity in his T cells, we find that alpha-synuclein specific responses are higher pre-onset of clinical Parkinson's disease compared to after. And here we're looking at a period of approximately 20 years, and we're measuring the immune response that the T cells make. And in the dotted line, you can see a control, so that shouldn't change over time. And as you can see, it remains constant. When we look at the alpha-synuclein, we have higher reactivity here prior to onset than compared to after. And you can see there's a huge amount of variety in the middle here when the individual experienced shingles. But when we summarize this in the immune response pre-onset versus after, you can see again that there's higher reactivity pre compared to after, and the control don't change. So that's very interesting to us, because it seems that there is a variation in T cell response. So we then moved on to a cohort of Parkinson's disease patients, which are at different times from diagnosis, because of course it's very difficult to have an individual followed for a period of 20 years. So we recruited a group of individuals with Parkinson's disease and again measured their immune response to alpha-synuclein. And here you can see that the closer you are to diagnosis, the higher reactivity you have. So there's more patients here that have an immune response against alpha-synuclein compared to a period further out from diagnosis. And we confirmed this in 2 independent cohorts, and research is always important to be able to confirm your findings. And we also didn't see any correlation with MoCA and UPDRS. So MoCA is a cognitive test that is used to assess cognitive function, and UPDRS assesses motor function and are commonly used in Parkinson diagnosis. But the T cell reactivity does not correlate with these parameters. So we published it and think that peripheral T cells are realistic models to study Parkinson's disease and progression of disease. So what about synuclein before diagnosis? So now we've changed the axis to look at specific T cell reactivity. And it seems if we take our 1 participant example, that we have higher reactivity in the prodromal phase, and then it declines in the clinical phase, which we also saw in the Parkinson patients. So we want to study this in more detail. So can we detect these responses in a large cohort? Can we use it for easy and accurate diagnosis? And if these T cells cause the disease, are we able to block them and actually prevent disease or the decline of neurodegeneration? And that's where our newly funded program comes in, which is ASAP. And the goals of this program is really to support diverse groups of international research teams to look at Parkinson's disease development and progression. And we're fortunate to be part of a team with very skilled researchers. So our collaborators come from New York, so Columbia University, Alabama, Caltech, and Montreal for this particular project. And we're very excited to be working on this, answering all the questions we have in Parkinson's disease. So our goals for the ASAP project is to look at this, exactly this. How does the alpha-synuclein-specific T cells change with disease progression? Are there any missing targets? You've heard us talk about alpha-synuclein now for the past 20 minutes or so, but are there other proteins that might be involved in this response? And is there a general inflammatory status that Alex was touching on in his talk to make the T cells be hyper-responsive in Parkinson's disease? And it has been shown that individuals on particular immune suppressive drugs, such as anti-TNF, which dampens inflammation, actually has a decreased PD incidence. So this is given to individuals with certain bowel diseases, and it's been shown that they have a decrease in PD incidence. So that might be a tie to the disease development and an indication that the immune system is very important. So we use a lot of patient samples. For ASAP, we're gonna focus on individuals with high risk of Parkinson's disease. So rapid eye movement or REM sleep is associated with dreaming. And individuals with REM sleep behavior disorder, or RBD, are acting out their dreams. And those individuals are at high risk for Parkinson's disease. So they can be followed and look at T cell responses over time. So what we'll do, we'll recruit individuals and follow them over time. So here we have the same individual followed over a period of few years, and obviously many people. And we will look at also Parkinson's disease individuals at the same time, and we'll collect clinical parameters, prodromal symptoms, and also vaccination history, so that we can see how T cell reactivity correlates with these parameters. And then we'll go on to measure specific immune responses. We use oftentimes this type of assay where you see each dot is a cell. So we're able to quantify the response by counting all these dots. So we'll measure T cell responses against neural antigens such as alpha-synuclein, but also others that might be involved. Different vaccine-derived targets such as tetanus, pertussis, or flu, and also frequent exposure targets to see if there's a general higher response in Parkinson individuals compared to their controls. So ASAP is very complicated and the whole project is quite involved. We will be involved here looking at clinical samples and T cell reactivity in individuals. We also have a group looking at mouse models so that we can really look at immune activation outside the central nervous system. Also look at gut pathology and how the gut may play a role in the initiation of Parkinson's disease, and then how it spreads to the central nervous system and the brain, looking at all types of different cells and ultimately in the brain itself. So all our success relies on patient samples and the generous participation. And we're very lucky to have such great collaborators, both local and also across the US and Canada, where we have Alabama, Columbia, Montreal, and Shirley Ryan in Chicago. And we really rely on access to viable cells so that we can look at what they do from these blood samples, and that relies on controlled temperature and time. And I will show you in the lab a little bit later how that happens. So we're always looking for participants to get involved. So here you can see some contact information where you can contact us to join our research study. So really, we're very interested in autoimmunity and the role of T cells. And we think we can use this to improve the diagnosis of the disease, unlock the key so that we can interfere early before all the neurons are gone. We're also looking at finding gene signature that contributes to the immune response, which will also help with these aspects, and really ultimately understand the progression of the disease so that we can stop Parkinson. And with that, I will head into the lab, and bear with me while I transition. Okay. So when we get our samples, they arrive in this type of box. And I mentioned that it's very important to have a controlled environment. And particularly for blood samples, we need them to be at room temperature. And of course, they're shipped from across the country. So time is also of the essence. So we need to get this box from within 24 hours from the collection of the blood. And this box is great because it allows us to keep the temperature constant for that period of time. It has these insulating materials that keeps everything at room temp. Our samples are shipped in these biohazard bags that are sealed. And when we get them, they're also wrapped in absorbent material because, of course, if any leak happens, you don't want it to go all over the box. So we just collect in these blood tubes, the regular tubes that are used in doctors' offices. And when we get them, they're put through a series of centrifugation steps so that we can separate the white blood cells, because those are the ones we use for our studies. And once we have them, the white blood cells, I dig around in my pocket here, we stick them in these cryovials, which will preserve them then in a special type of media. Once we have them here, they go in these types of box. As I mentioned, temperature and time is always crucial, and this box will allow it to freeze at a controlled setting so no cells are left hurt in the process. And these boxes actually go into the minus 80 degrees Celsius freezer, which you can see here. And they sit in here overnight so that they have a controlled freeze to keep the cells as happy as we can. And once they've been in there for 24 hours, we want to bring them into our liquid nitrogen tanks. And I'll show you what that looks like. Of course, we need a bit more safety precautions as these go down to minus 180 degrees Celsius. So it's very, very cold. So we need some insulated gloves because you don't want to get any freezer burns from the tank. And it has this huge lid that of course is very insulated to make sure everything is kept cold and frozen and not too much leak. And we stick our vials in this type of rack in these types of boxes. And there they can stay until we need them for our experiments. So this is really great, and it keeps everything preserved and happy. So there you have it. That's how it works with our different blood samples and how we get the cells we need for our experiments. And with that, I will hand over to Steve, and I will see you for the question and answering portion.
Thanks, Cecilia. Everyone, before I get into questions and answers, I have another important segment that I'd like to talk about. So the La Jolla Institute for Immunology is a basic research institute. It's a translational institute. And each year we compete for the funds that provide research support. In fact, about 70% comes from annual competitive grants and contracts. And that shapes the studies that we're able to perform. We also partner with industry, which comes with a set of priorities themselves. And the last and very important component is philanthropic giving. Philanthropy is consequential, and it's a powerful affirmation of our mission to serve the community. I mean that sincerely. It tips the scales in the direction that donors like you choose. Donors have their priorities, be it Parkinson's or others. And that funding is very, very catalytic in terms of getting the studies in those directions. And this is good. We're scientists, but I have a dear friend with Parkinson's. And so this is personal as well. And so we like the chance, we want that chance to be able to take risks and study areas where immunologists can have an impact. I can tell you that philanthropic funding here at the Institute is extremely elegant as well because of a couple characteristics that are different than grants. It's fast and efficient. Oftentimes, we and donors are able to discuss the use of funds, be it for general research or for specific diseases, and immediately go into action. We're able to take risks, and what I mean by that risk-taking is to go outside of dogma or go outside of established programs at the federal level that may have certain criteria for funding. Instead, we can go right at something that's high risk, high reward. I think some of the evidence there is when Alex mentioned that just 3 years ago, these studies began, and an immunologist study, a study of immunology immediately had these earth-shaking changes to how we look at Parkinson's going forward. And finally, philanthropic support is one of the only ways that the Institute can have long-lasting support. Our grants are spent as they're gotten, but philanthropy can make a lasting gift so that we can pursue the challenges all the way through to the end and get that capital equipment, recruit those scientists, but really chase down problems to their conclusion. Donors and philanthropic support is really what ignited Parkinson's research here at the Institute. And you can see that it's been very well stewarded. Here at LJI, it's provided advanced funding to get the reagents that are specific for studying Parkinson's, to set up the systems that were built for other studies to look at Parkinson's disease. And more important, it's created a rugged program that here at La Jolla Institute, we will be studying Parkinson's until there's a solution. Neuroinflammation has become integral to what we do at the Institute. So if you're interested in Parkinson's disease, or are now even more impressed with the power of the immune system and the study of it, I encourage you to consider giving to La Jolla Institute. Now with that, let's switch gears a little bit and get into some questions and answers. I have a bunch. I'm going to try to balance it out. And I pulled one here. to start with. So Mitch, are you ready?
I'm ready, and they're gonna let me start my video.
All right.
Okay,
I'm
here I
going
am.
to start with you.
Okay.
The question is, you talked about autoimmunity playing a role in Parkinson's disease. Could immunosuppressive drugs used for other autoimmune diseases, such as Humira or Enbrel, be used to treat Parkinson's?
Well, that's a hypothetical possibility. One of the problems is the long timeline of the development of these diseases. Another problem with the case mentioned, those are what's called biologics, and they have trouble getting across the blood-brain barrier. Now, people are trying. There's a company, for example, in San Diego called Armagen, which has been trying to develop a TNF blocker. That's what Humira is. And they're trying to develop one that can cross the blood-brain barrier. So there may be ways to get these molecules into the brain. But then if we return to the problem of the long time needed for treatment, the immunosuppressive effects of these treatments, and finally, the fact that on repeated treatment, patients tend to develop resistance to these molecules, in part because their immune system sees them as foreign. So I don't think that's an easy solution, but it's probably worth continuing to explore.
Great. So this is a bit of a follow-on, but there's kind of a theme to some of the questions that we're getting. So I'm going to incorporate this. Cecilia and Alex, are people who are on immunosuppressive drugs, in your thought or data, are they less likely to get Parkinson's?
So I can take that, Steve. We haven't done those studies ourselves, but as I mentioned, individuals on certain types of immunosuppressive drugs, those that dampen inflammation, actually have been shown to be less likely of developing Parkinson's. But like Mitch mentioned, I don't think it's the solution to put everybody on those biological drugs to prevent the disease. So, of course, a lot more studies are needed, and we hope that our work will contribute to answer those questions and also help identifying individuals that are at high risk of developing the disease, so that something can be done early enough before all the neurons have degenerated.
That's great. You know, Alex, there's a question that I suspect that a lot of people are wondering, and I'm hoping that with both Parkinson's and other studies that you've been doing over the years, you can help answer. In your mind, is there data to suggest who from a genetic standpoint, so rather than just men and women, but ethnicity, nationality, regionality, environmental factors, I suppose, can come in, but really on the sort of global basis, are there populations that are starting to emerge that may have an inherent genetic risk besides sex differences to Parkinson's?
Yes, so certainly apart from the gender bias, which is well known, males tend to be more susceptible to developing Parkinson than females. There are a number of mutations and genetic traits that are associated with increased risk of developing Parkinson. In general, those have been hard to study from our point of view because they are rare. So it's not like 80% of a Parkinson patient have one particular mutation. There are these particular mutations that are frequent. They are also frequent in particular ethnicities, but very specific. So yeah, so it clearly, Parkinson is almost a series of diseases that, or a disease that can be triggered by different biochemical pathways.
Right. So I'm going to take one, but after I answer, actually, everyone should feel ready to jump in. It's a bit of a follow-on to what was introduced in the talk, and the question is, do we anticipate a wave of Parkinson's disease as a result of the COVID pandemic. And so I saw this and thought about it. Now, thanks to some basic research on the brain, we actually know that the receptor that COVID uses, ACE2, is present and can infect those cells in the substantia nigra. And since viruses invade cells and then the immune system needs to go and destroy them, as the talk I think very elegantly showed, that causation starts to come through. But in terms of a wave, first, I hope the talk illuminated the fact that Parkinson's is something that is generated over time. And for context, human beings have pre-existing conditions from the moment we're born. We're infected, and many things infect us. And some do get into the central nervous system and are catastrophic, but others are not. So I think that this question really shows why this work is important. I think it's also, I'll just put in a parting note that, in fact, if many people are asymptomatic for COVID, screening is very important as well, so that people will know their history of it. And finally, it's a plug for preventative medicine again in developing vaccines, even if the virus and the respiratory disease weren't the worst thing to worry about, the things that come in. So my opinion, and I'd like others on the other panelists to comment, my opinion is we wouldn't see a wave immediately, though the prevalence could be impacted, and research here is going to really point to it. So please.
Right. So there's really not a lot of data at all. There's a couple of papers that came out, but really single case studies, so 2 people, where they have reported that someone had COVID and precipitated what appeared to be a Parkinson. I am on the fence on those reports because, again, Parkinson is difficult to diagnose. It's well known that one of the side effects or lingering effects of COVID can be alteration in the brain, and people suffer neurological things. So I don't know whether that was really Parkinson. So it's kind of a little bit. And I certainly haven't seen a wave of Parkinson. The other point that I was trying to make earlier is that it cut both ways. It could be that Parkinson have their own higher level of exposure to viruses that could have potentially a protective or ameliorating disease. So it could cut both ways. And so we've been very interested in, actually, have been trying to develop some epidemiological data. And we have been interacting with all our collaborators and say, so do you have any case of someone that had Parkinson and got COVID? And if you do, we want to study them. And we have been able to, so far, to find any case, which again, maybe is consistent to the reason why you can't find them. But it's a very complicated thing to do, because if you see that Parkinson, the incidence of COVID is lower in Parkinson, that doesn't necessarily mean that they're protected. It means that, for example, Parkinson patients may have a more in place, more sheltered life. They don't go out as much, so that the chances of being infected, especially early on in the pandemic, might have been lower just for lifestyle issues, not for epidemiology. So we're trying to understand it, but it's not it's not entirely straightforward.
So Cecilia, I was going to ask, given that children are not currently a part of clinical trials and are largely symptomatic, if you look at statistics, when you think about Parkinson's and the long timeline, do you think that we're missing out Or do you think that your data will show us the value of vaccination as a preventative measure? I realize it's speculative, but just your thoughts.
Yeah, I think it will do. I think it never hurts to get vaccinated for a wide variety of reasons, right? You're protected against that particular disease. And yeah, I think that's a great thing to do. If it has any contribution to help against Parkinson's, that would be a great side effect. But you should get vaccinated based on being protected from all those diseases and to protect those around you.
Thanks, Cecilia. So
yeah, certainly I want to echo that in terms of it just the flu vaccination, right? So certainly I get vaccinated every year, but even this year, even more so, because this is not a good year to get a disease that looks like COVID. I mean, you want to avoid having the angst and the issues or having to be hospitalized, God forbid, with the flu. It's paramount to do that. Also, I just wanted to go back, since we're talking about vaccination and flu, another interesting aspect that we're, at least interesting to us, but we want to look at. So we've been discussing, so that maybe Parkinson is linked to some kind of generalized inflammation, right? Mm-hmm. This is, such a trivial question that has not been answered. So, for example, what if you take 20 Parkinson patients and they get vaccinated against the flu, and then you take 20 controls? Do Parkinson people, because they have a heightened inflammation, do they respond better to the flu vaccine? Do they respond less well to the flu vaccine? It would seem, but it's such a basic question, but that would be answered. It's not been answered. And in fact, our understanding from our clinical collaborators is that when the Parkinson patient usually goes to see their doctor, the doctor will offer them, do you want to get the flu shot? And usually a large fraction of them, they will. So it's not like we're doing anything strange, but the information is there, just interesting to extract that.
It is, and I think this is the immunologist's perspective on what was long a neurologic disease. So, you know, Mitch, I wanted to go back because there were a couple of questions around it, and have you talk a little bit about why men are more, or better yet, why women are less likely to get Parkinson's disease. Does it have to do with their immune system? Is this known?
It could. We know that men and women have very different immune responses. And some of that may be due to hormones like estrogen. Generally, women tend to be more susceptible to various kinds of autoimmune diseases. Probably one of the most striking examples is lupus, which is 90% female. In other cases, and Parkinson's is one, actually COVID-19 is another, men are more susceptible. And we don't understand all the reasons. We can't rule out lifestyle issues. Perhaps, you know, men are more subject to head injuries from various kinds of sports, although that may be changing as society changes, or may be exposed to more toxic chemicals in the workplace due to their work history and so on. So we can't rule out the sociological factors, but there are definitely strong biological differences between the sexes in immunity, and it's an area that really does require, another area, if you will, that requires further investigation.
Agreed. Cecilia, a question was, you know, can inflammation in the gut lead to Parkinson's? And you talked about studies that will include gut pathology, but could you elaborate just a bit more?
Yeah, so that's a very interesting question. We've had studies that points towards the gut that we didn't do ourselves, but other investigators, where it's thought that particularly the appendix has the same protein aggregating, so alpha-synuclein aggregate. And it's believed that somehow the pathology spreads from there to the central nervous system. And that's really something we'll be able to investigate with the ASAP funding and really tie that together with our observations in synuclein-specific T cells and other targets that we will study. So I think it's a really interesting avenue of exploring. I don't think we have all the answers. Hopefully we will in a few years' time. But yes, there could be a contribution of inflammation in the gut towards developing Parkinson's disease.
Great. And this next one I have is for all of the panelists. And Cecilia, since you just answered, rather than rapid fire you, I'll ask Alex or Mitch to ask first. And that is, in thinking of Parkinson's now differently and the immune system's involvement, is it thematic? In other words, is Parkinson's one of other central nervous system diseases, let's take Alzheimer's, for instance, that you think will find similarities in either how they start or how the immune system is an integral part? What are your thoughts and maybe even some data that you think is compelling?
Alex, go ahead. Alex, why don't you go first?
I think there are certain traits that are common. So one thing that is strikingly similar in many neurodegenerative disease is this business of having some clumps of some protein. And of course, the amyloid plaque is characteristic of Alzheimer's, synuclein, Parkinson. There are other instances of that. So that is a common trait. On the other hand, in terms of the T cell and immune reactivity, it seems that they are different. Actually, we just published a paper, Cecilia and I, where we looked at a first look at Alzheimer's by the same approaches that we had used for Parkinson. There, we didn't see as many differences as we saw in Parkinson. So it could be very, very different on a case by case.
Yeah, I think it'd be very hard to generalize, as Alex said. One thing we've learned in recent years is that the brain is subject to a greater degree of immune surveillance than we realized, and also that peripheral neurons secrete chemicals that communicate with the immune system, various types of cells. So the connections there are probably much deeper than people realized even a few years ago. And that will help us to understand neurodegenerative diseases of various kinds.
That's great. So I'm looking at the time. It's just about that time to wrap up. I wanted to ask if the panelists had any parting thoughts. Otherwise, I was just going to point all the attendees to our last slide. And I have a couple of important reminders. So the first is there will be a recording of today's webinar. So if you want to share it, you'll be able to do that. On screen, you can see various ways to contact the Institute for both donations as well as getting involved in our studies. I mentioned to you that Parkinson's is a very personal thing to me. I'm sure it is to many of you. And we're always looking for people who will help move the science forward. And then finally, a last and sincere thank you. Parkinson's and the work here has been particularly impacted by philanthropic gifts. And we think that it's this kind of funding that is actually going to let us make some real leaps here. And so I hope that I've convinced you that even though it's the La Jolla Institute for Immunology and Parkinson's may not have been something that you or friends have thought of as central to Parkinson's, your immune system is very much in the middle. And so with that, I would just like to again thank you and remind you that we'll see you in November, our next Live from the Lab webinar. And considering the times, details will follow as to the topic of choice. So again, thank you, and get in touch.